No, that's not what I wanted to share. Huh, that's interesting. I don't know what you guys see, but that's the midterm exam. Let's try this. There we go. All right. So today, like I said, we'll be talking about microbial controls, physical, chemical, mechanical controls. To give you a little overview of the chapter, we'll talk about the different techniques and how they differ from each other. How is sterilization different from disinfection and antisepsis, for instance? We'll talk about some of the methods employed to do this. This is an image of an autoclave. We have an autoclave in the biology lab, and if you guys were doing a face-to-face lab, we would be having you personally use these autoclaves to sterilize your media, so your agar and your broth and some of the equipment. We'll talk about the actual mechanism of action, so how these disinfecting and antiseptic agents take down bacterial cells and yeast cells and other fungi and viruses. We'll talk about the wide range of chemicals that are out there, these germicides, and some of their different degrees of efficacy, and whether or not they should be used on inanimate objects. So like spraying Lysol on a counter is okay. Spraying Lysol in your mouth is not okay. You want to use Listerine, right? But using Listerine or a mouthwash on a counter probably isn't that, effective. So we'll talk about these different chemicals and which ones are appropriate for different things and their degree of microbial control. We'll go into the different categories of chemical controls. So this can be halogens, alcohols, peroxides, aldehydes. If you're taking organic chemistry or you've taken it before, you'll be familiar with a lot of these different classes. And that's it, right? So when we talk about controlling microbes, we can do that physically. So think of things like temperature control or irradiation. We can do it chemically. So think of things like alcohols or bleach. Or we can do it mechanically. So like filtration. And that is the way that we either destroy or reduce the load of microbes for any given sample. And that process is called decontamination. And the primary targets are microbes that cause either infection or food spoilage, right? So we want to protect our food. We want to keep it lasting as long as possible. We also don't want to get sick. And so these microbial agents are going to be things like bacteria, fungus, like in this bread. This is black mold on bread, viruses, obviously, and protozoa, things like that. Algae, not so much, right? Although there are microbial controls that you can put if you have a pond that you don't want to have it turned into just an algae looking cesspool. These different controls you need to know because they are different in the ways that they're used and the degree in which they kill microbes. So disinfection is something that destroys the vegetative cells. So remember, when we're talking about bacteria, you have vegetative cell, which is the metabolically active type, and then the endospore, which is the metabolically inactive type. And so disinfection is going to take care of pretty much most of the metabolically active vegetative cells and viruses and fungi. But it's not going to take care of endospores. So it's not sterilization. And disinfection we use on inanimate objects. So you don't, semantically, right? You don't disinfect your body before going into surgery. Okay. You disinfect your counters after you chop, raw meat or something. Sterilization, in contrast, is the complete removal of all viable microbes. Okay, so that is taking care of the endospores as well. Nothing should be able to grow after you do this process of sterilization. Also used on inanimate objects. Okay, you cannot sterilize your body. It just isn't going to happen because the conditions needed to sterilize microbes will also, harm you as well. So that brings us to antisepsis. That's the process of removing microbes from an animate object. So when you are applying an antiseptic to something, you're applying it to your body, okay, not the counter. And this is going to reduce the microbial load. It's going to destroy or inhibit the vegetative pathogens. It's not going to get rid of endospores, though. Okay. If we don't kill things, we can also use a method called microbiostasis. So stasis means static, right? Same, it stays where it is. And so rather than just killing off the bacteria or whatever microbe you're wanting to reduce or control, microbiostasis is to just keep it at bay so they no longer can replicate to get to higher loads. And remember, when we talk about infection, we said that there is, a specific dose, a specific number of pathogens that are required for each microbe to take up infection, right? And so if we can prevent small amounts of microbes from growing into larger amounts of microbes, then that's going to reduce the spoilage of our food. It's going to reduce the ability for them to infect us. And so what's one way to control microbes? Anybody know what a form of microbiostasis might be?
Is antibiotics one?
Antibiotics can be, although antibiotics are largely, hopefully, a microbicide. So it's going to hopefully not just keep it at bay, but kill those microbes, which is why you want to take, again, the full course of your antibiotic regimen, because you want to kill off everything and not leave some lingering to have adaptations and mutations that can then make them resistant to antibiotics. I heard somebody else saying something.
Maybe pasteurization?
Pasteurization is a good guess, and we'll talk about that, but that is a microbicide. It's not a sterilant, right? So it doesn't sterilize everything, but it does reduce the load. But it does that by killing off a certain amount of the pathogens. No, what I want you guys to think about is refrigeration. So why do we put things in a refrigerator? Because the temperature is such that it doesn't allow most microbes to divide and to grow. It doesn't kill them per se, but it's not a hospitable environment that allows them to flourish. So when we put things in our refrigerator, they still go bad, right? If you give it long enough time. But it slows the rate at which the food goes bad. So that's microbiostasis. Now, methods that can reduce the number of microorganisms can be things like sanitation. So we're removing, again, these microbes from inanimate surfaces. We're not killing off everything. We're not sterilizing it, but the idea is that the less number of microbes on that surface, the less number of microbes that get onto our food or into our bodies. And then degermation is reducing the load from living tissue. Okay. So sanitation, inanimate. degermation, animate, disinfection, inanimate, antisepsis, animate. So these terms you'll need to know because they apply specifically to different methods and different targets or different subjects that are being reduced in microbes. And so here's a list. You don't need to memorize this, but this gives you all the terms that you'll need to know and gives you examples and what their application is. Now, when we talk about the resistance of microbes to these controls, there's a range of things that are highly resistant to things that are not as resistant. Prions, and as we've talked about, endospores are very highly resistant to microbial controls. In the middle zone, you have things like protozoan and naked viruses. So what's a naked virus? It doesn't have an envelope. And we talked about them being more stable than enveloped viruses. Bacteria that don't produce endospores, but have a nice thick cell wall are going to be moderately resistant. So some of these things like Mycobacterium that causes tuberculosis or Pseudomonas, these gram-negative are going to be moderately resistant. Staphylococcus is going to be heat and chemically resistant. Staph aureus is also, right, antibiotic resistant, methicillin in particular. And then when we get to the least resistant types of microbes, now we're talking about the vegetative cells of bacteria. We're talking about the spores of fungi and the hyphae, the little branches that they shoot out. Yeast, which is also a fungus. Enveloped viruses, they're much less resilient than naked viruses. And then protozoan trophozoites, so you know, the eukaryotic microbes that are like the malaria parasite, for instance, and other ciliates. Okay, any questions on this so far? Now, one thing that affects the rate in which you kill microbes is time, right? So if you have time on your side, you'll eventually kill stuff off, right? So we look at this right here. We have this flask and it has a number of microbes in it, and we're giving it some microbial control, whether it's heat or a chemical. And over time, it's going to kill the microbes in there and you're going to have sterilization, right? But there are other things that influence how much time is required, right? So if you have a high load of bacteria, for instance, so you have a lot of viable bacterial cells, it's going to take more time to kill them off than it would if you had a lower load of bacterial cells, right? Which should make sense. If you are dealing with microbes that make spores, right, those are more resistant. And so it's going to take more time to kill those off than it would to kill off the vegetative cells. And then finally, it depends on the type of agent you're using for control. Okay, so here, if we are using a microbiostatic agent, then you're not going to kill off anything. You're just going to keep it at bay, right? The population just levels off. However, if you're using a microbicidal agent, then that will lead to a decrease. Okay, so these are just different factors that will influence the amount of time required to kill off these microbes. Most things are a function of time in general. How do these control agents actually do their job? Well, a lot of them break down the cell membrane of the bacterial cell or the fungal cell, or the capsid of a naked virus, or the envelope of an enveloped virus. So they have so if whether it's a particular type of chemical in particular, it's going to have these surfactant molecules that intercalate into the membrane and then cause that membrane to start falling apart. And remember that the membrane is like the protective coat, right? So if you start breaking down that membrane, it's no longer selectively permeable, it's completely permeable, and the cell basically disintegrates. That's a common mechanism of action for a lot of chemical agents that we'll talk about. Another way to destroy pathogens or to destroy microbes that cause food spoilage in particular is to denature their structural components, right? So we're taking the protein that has a very specific shape, and we've talked about this, a lot now, that a particular conformation of a protein designates a very specific function for that protein. And so you can either denature it to where it now has a different shape right here, using, for instance, heat, or changing the pH, if it's not a thermophile or if it's not an acidophile. Once you change the shape, it no longer has the same function, and then that function is no longer available to that microbe, and that might be a very important function, and so that the microbe cannot subsist after that. You can also, for instance, when we'll talk later about heavy metals, those heavy metals can actually block certain active sites on the protein, and now that substrate that is looking to bind cannot bind because it's already being blocked by something else, again rendering that protein functionless. If you give it enough heat, you'll just completely change the shape to where it's just loose, and now it can't do anything either. Okay, so we can break down membranes and walls and barriers. We can denature proteins. We can also inhibit the synthesis of these biomolecules from the start. So before that protein is even made, some of these agents can get in the way of replication, in the way of transcribing RNA, of translating that RNA into protein, getting in the way of peptide bond formation. So, interfering with the ability of these peptides to form proteins. And later on, not today, we'll talk about the use of antibiotics and how they work. And a lot of those antibiotics that we're familiar with are functioning at this level. They're getting in the way of synthesis. Okay. Any questions on that? That makes sense? These are the major modes in which, from at least a chemical aspect, we control microbes. All right, so real quick check. If you wipe your hands with an antimicrobial cleaner, what is that? Is that sterilization, disinfection, antisepsis, or sanitization?
Would it be C?
D.
Sanitization.
I heard C. I heard some other things. The correct answer is C. Because remember, sterilization, you can't sterilize your body. That's killing off everything, including your body, if you were to go through that process. Disinfection is to control microbes on an inanimate surface. And sanitization is also, even though we use sanitize colloquially in an improper fashion, sanitization is also controlling microbes on an inanimate surface. Degermation, if that was on here, that would be an option. So antisepsis or degermation would be options. Now, when we are deciding which microbial control to use, we have to think about what we're using it on, right? Does the application require sterilization, for instance, right? If we're using surgical equipment, we want to make sure we sterilize it, right? You don't want to just wipe out some of the microbes. You don't want to do microbiostasis with that. You want to kill off everything because that's going inside the body where things should be sterile. Is the item to be reused? So in basic research, most of the stuff that's being used is plastic nowadays because you can throw it away. Horrifically so. Everything is plastic. Because, you know, it's much easier to just throw it away than have to re-sterilize things. So if the item is to be reused, it's probably not going to be plastic if you're going to sterilize it, because a lot of plastics don't hold up to the sterilization process, the high heat and the high pressure required. Which is the third point, right? Can it withstand heat, pressure, radiation, or other chemicals? Certain chemicals will break down certain materials, right? So you got to know what you're using and what its tolerance is to these different controls. Will the agent penetrate to the necessary extent, right? So if you just want to sterilize the surface of something, that's going to be a different question than if you need to sterilize or control the microbes deeper into that material. Radiation, for instance, certain types of radiation have a deeper penetrance, right? And so other types of radiation can be stopped by a very simple barrier, right? And we'll talk about that in a little bit, the difference between ionizing and non-ionizing radiation. And then, you know, practically speaking, is this going to be cost and labor efficient and safe, right? So if you're wanting to decontaminate something, you don't want to pick a control that's going to be highly cost prohibitive, right? Or something that if it's a consumer product, that it's going to be unsafe for the users, right? So just some considerations. Most microbes are readily controlled by changing their environment, unless they are those extremophiles that we talked about. But most of them have adapted to a very particular niche that we talked about last week, right? And so if you change that niche, the conditions of that niche, you change the ability of these microbes to survive, whether it's heat or it's the lack of heat, or it's drying out the environments, or it's irradiating the microbes themselves, or it's filtering by removing the microbes. All of these things are ways in which we physically control microbes. And we'll talk about chemical and stuff later. But these physical methods are, as I said, heat. And heat can be in the form of a dry heat, so incinerating something or having a dry oven. Or heat can be, and I hate this word, but moist. It can be a moist heat. And so from now on, now that I've said it, we'll just say a sufficiently hydrated heat. Now, this is what an autoclave does. This is how we sterilize things in the biology lab. So you have a steam, a pressurized steam, and that pressurized steam in conjunction with a high temperature is greatly effective at killing off the endospores to sterilize. Pasteurization is also a form of this sufficiently hydrated heat. Radiation, ionizing, using X-rays, gamma rays, that's going to sterilize. Non-ionizing is things like UV. That is a disinfection, so different, right? Non-ionizing radiation will not sterilize something. It will disinfect it. So those UV straws and stuff, that's not killing off the spores. Okay, that's just killing off most of things. Another way to physically decontaminate something is by a mechanical method, filtration, right? We filter air, right? Your heating ducts have filters. Your cars have air filters. That's a disinfection, though, right? Because depending on how, depending on the size of the pores in that air filter, certain things can still get through. However, if you filter liquids, typically we have membranes that are small enough to trap most microbes. Then we get into chemical agents. There are different gases that can be used, chlorine gas, liquids. And all of these things have different targets and different degrees of control. Okay, and that's what I want you guys to get out of this. If we're filtering air, that's not going to be sterilization. Liquids, yes, right? If we're using liquids on animate things, that's going to be antisepsis. Inanimate, it's going to be disinfection and potentially sterilization. Okay, does that make sense? Okay. Any questions so far?
Yeah, I have a question.
Mhm.
You said that changing the niche conditions will typically kill the microbes unless they're extremophiles. So if they are an extremophile, just changing their physical condition won't actually kill them?
I mean, it can.
Okay,
The thing is with extremophiles, they're typically not going to be interacting with us, right? Because they live in extreme conditions.
okay,
We don't worry about them on our food or on our hands.
gotcha.
So the conditions that so what you would do is if you changed an extremophile condition, it would be changing it to a condition that's hospitable to us. Okay, and that would probably destroy it as well. So if you take something that is used to thriving in a very hot environment and you put it at room temperature, right? So a hyperthermophile, and you make it you give it a mesophile condition, it may not like that. So thank you for that clarification. I don't want to confuse you and think that extremophiles can necessarily exist in any kind of environment. It just means that they exist in these extreme environments. And those extreme environments aren't things that the microbes that we're trying to destroy, they don't encounter those environments. Does that make sense? Maybe? Okay.
Absolutely. Thank you.
Okay. I can't see anybody because I just pinned myself on this so that when I post this online, if anybody doesn't want to be seen, they don't have to. So I can't see any of you. I'm lecturing to myself right now unless you say something. Okay.
Okay, sorry, I was nodding vigorously. I didn't realize.
Yeah, exactly. Okay. So again, sufficiently hydrated heat, you can use that at lower temperatures and shorter exposure times. And what that does is that denatures the proteins so that you no longer can, the microbes cannot undergo the metabolic processes they need to stay alive. Dry heat, you need to be higher. So you're going to incinerate stuff. By doing that, it will denature things as well. But it also dehydrates, it evaporates all the water. Water is necessary for life. And so those are the contrasting features between sufficiently hydrated heat and dry heat. So when we talk about the sufficiently hydrated heat, sterilization with steam under pressure, that is autoclaving. And that's usually 15 psi. You do have to adjust your autoclave at altitude, but we typically don't. It's still effective at 15 psi, but 15 psi at 121 degrees Celsius for usually about a half an hour, anywhere from 10 to 40 minutes. And the things that you use in an autoclave, of course, can't be sensitive to heat or to moisture. Okay, so if you're wanting to sterilize metal, like in terms of metal equipment for surgery, an autoclave may not be the best thing, because over time that can corrode the metal by getting it wet, right? So you might want to use a dry oven for metal objects, but glassware works great for this. Non-pressurized steam is not autoclaving. That's going to be tyndallization, which is kind of a fun word to say. That is going to use this intermediate process for substances that cannot withstand the harsh treatment from an autoclave. Okay, so you expose it to steam for 30 to 60 minutes, and then you incubate for 24 hours, and then you subject it to another round, and you do that for 3 days. Okay, and so this would be a process that might be used for things like canned food, right? If you autoclaved canned food, you would definitely sterilize it, but you would also be destroying all the nutrients in that can, right? And so pasteurization is one way to do it, and then tyndallization is another way. Boiling water, of course, that's a sufficiently hydrated form of control. That's disinfection, though. 30 minutes at 100 degrees will destroy most pathogens, with the exception of spores. Pasteurization, right? We talked about that when we did the that Labster demo. You apply heat, but you pulse it, right? It's like a flash. High heat, 71, not 121, 71. Not for 30 minutes, for 15 seconds. Okay, so it's a quick burst, doesn't sterilize, it just decreases the amount of microbial diversity and life in that object. And then you combine pasteurization with refrigeration for microbiostasis, and together you have a longer shelf life of a product. Okay, keep in mind, this doesn't kill endospores. So there is a process called ultra pasteurized. And so that you might see that on certain products. That is when you take it at 134 degrees Celsius for 2 to 5 seconds. Okay. So we're going up higher than autoclaving, but not for the same duration. And I want you to know these different procedures. Do I care if you know that it's 134 degrees for 2 to 5 seconds? Not so much. What I want you to know is the difference between pasteurization, tyndallization, autoclave, ultra pasteurization. Okay, know that ultra pasteurization is much higher temperature than pasteurization, shorter period of time than pasteurization. Okay. This brings us to the dry heat incinerators. So we actually have some of these things in the lab. We typically use Bunsen burners, but this is a more safe and more effective way, where it just has this really high heat. And this person here is using their inoculating loop, and they're sticking it in there to kill off the microbes instead of using a Bunsen burner. And then the hot air, or the dry ovens. This is going to be, again, hotter than the autoclave at 121. It's hotter. And it can be longer too. It can be anywhere from 12 minutes to 4 hours. Okay. And what it does is it coagulates the proteins, or it basically denatures them, and then they just kind of stick together gloopy, and they can't carry out their function. Another nice thing about the incinerator is that it keeps everything inside there, right? So if you use a Bunsen burner and you have, say, some broth on there with your microbes in there and you put it on, it could sometimes crackle and spatter and then fling stuff out. So this is a more contained method of using to decrease microbes on your lab equipment. Microbiostasis, as I mentioned, lowering the temperature. So refrigeration, freezing. This is how we control microbes. We don't disinfect the microbes from our food. We just control the amount that are there so that no more can divide and then increase the rate at which our food spoils. Desiccation, that's just to remove the water. You dry them out, then they're inactive. And lyophilization, when you lyophilize something, you freeze dry it. Okay, and so that is a great way of preserving things as well. So you can get certain bone broth powder, or you just add water that's been lyophilized, so it's decontaminated that way. But these aren't good ways to completely disinfect or sterilize. These are just ways to keep microbes at bay. All right, another check here. Which of the following accomplishes microbicidal, not microbiostatic, right? Microbicidal kills. Which of these things kills microbes? So there might be more than one answer.
C.
Which of the following? C will not, actually. C is a microbiostatic process. Remember, if you decrease the temperature, then you decrease the ability of that bacterial cell, for instance, to divide. Same with desiccation, right? If you dry up something, you're going to prevent it from continuing to undergo cellular division. But that's not going to necessarily kill anything. It's just going to keep it at bay. Whereas boiling will kill. It won't kill everything, but it will kill a lot. Pasteurization won't kill everything, but it will kill some things. And incineration will kill some things, not necessarily everything. Sometimes it could, depending on the conditions. Okay, does that make sense, guys? So the first three are going to be microbicidal. D and E are going to be microbiostatic. Radiation, right? Radiation. We use radiation to sterilize things or to disinfect things, depending on the application. Ionizing radiation, that's the bad stuff, okay? That's the stuff further out from the visible spectrum of light. And so these are going to be gamma rays, X-rays, things that can penetrate deep into the tissue and break your DNA in half. Whereas non-ionizing radiation, that's a little less harsh. Ultraviolet radiation, right? So it's not going to break the DNA, but it's going to alter the DNA. That's going to lead to issues when that DNA is then transcribed into RNA. And we can go through this a little bit. So ionizing radiation is going to break the DNA. Non-ionizing is going to create these dimers with the pyrimidines, particularly the thymines, which then prevents replication because it's going to either kick off the polymerase when it encounters that dimerization, or it's going to not know what to do and just put some random nucleotide in there and introduce a mutation that might be beneficial, might be detrimental. Okay, so here's ionizing radiation. The radiation source can usually go through a lot of different barriers, depending on what that barrier is, and cuts right through the DNA. So you can see here, it breaks a double strand breaks the DNA in half. Gnarly stuff. Whereas non-ionizing radiation, will be stopped by most barriers, right? If you wear clothes, you aren't going to get a sunburn. And what it does is it doesn't break it, but let me get my annotation tool here. What it causes is it causes these, oh, that's bad color choice. It causes these nucleotides to bond to each other. Now, probably you guys all know the general makeup of DNA in that you have these nucleotides here. And this is a strand here. So let's say this is 5' end, this is the 3' end, and then this is the complementary strand. This would be 3', 5'. These nucleotides on this strand aren't bound together. They are bound to a sugar and phosphate backbone, right? And they undergo hydrogen bonding with the corresponding nucleotide of the other strand. So what non-ionizing radiation does is it causes these pyrimidines, these thymines that are next to each other to form bonds, which isn't supposed to happen. And so that like I said, that screws up the ability for the polymerase to then read that and see what corresponding nucleotide needs to go there for the RNA synthesis. Make sense? Okay. And that's how we get skin cancer, right? So if it does the same thing to our cells as well. UV, too much UV will cause the pyrimidines in our DNA to link together. And then when we replicate the genome in that skin cell, there'll be a mutation that's put there because the polymerase gets confused. And that mutation, if it happens to be 2 thymines in a cell cycle checkpoint, now it's mutated, and those cell cycle checkpoint molecules won't tell a cell now to stop dividing, and we get skin cancer. Effective uses of irradiation on things like produce. So we can irradiate our fruit, for instance, and 5 days later, there's no mold. Raspberries, as I hate buying raspberries. I don't know if you guys are the same, but I feel like I can never keep raspberries for more than a day or two, and then they look like this picture on the right. So I must not be buying irradiated raspberries, but of course it's irradiating the DNA in the raspberries too, right? It's but luckily they don't need to reproduce anymore. We're just going to eat it, and it doesn't matter if you eat dimerized thymines from raspberries. It's not going to harm us. Okay. Any questions so far on this? Okay. Filtration. So that's how we physically remove these microbes, by passing liquid through a membrane that has these pores. And the pores are so small that the microbes get trapped. They can't go through it. But water can. And that's how we filter out microbes from liquid. And in the lab, particularly, we have these filters, and then we put them, we put that filter on top of a flask, and we have some kind of vacuum filtration method to just force the liquid into that bottom flask, as it's quicker than waiting for gravity. And then all the bad stuff stays on the other side of the membrane. Okay. So we've talked about disinfectants. We've talked a little bit about antiseptics and how they differ from things that sterilize. We talked about degermers a little bit, and preservatives being things like irradiation. These are going to be all methods that are considered germicides. Okay. So a germicide kills germs, right? Hence the name. But they have certain characteristics about them, right? They can usually be highly effective in low concentrations. You don't need a very concentrated solution of hydrogen peroxide to kill things, right? And the stuff we buy is very, very dilute, the stuff you get at the store. Solubility in water, because you're killing life forms that exist in water, it's more effective to have an agent that also is soluble in water as a delivery system, right? You don't want germicides to be highly toxic to us, though, right? We want it to be bad for the germs, not that bad for us. We want it to be able to get where it needs to go. We don't want it to be corrosive or staining, right? If you're using this on your, again, these surgical equipment, we don't want it to corrode the tools and get them rusty. We don't want it to decolorize or stain whatever we're trying to disinfect, or if we're putting it on us, to anti-septic approach. And then we want it to be obviously cost effective and something that is not so rare that we can't get our hands on. Although in this day and age of COVID, it's been pretty hard to get things like at least initially, some of these germicides became kind of a hot commodity. All right, so at 10:51, we'll get into germicides on Wednesday. Any questions on this before I wrap it up? Before I wrap up the lecture? No? Okay, so pretty straightforward. Let's take a 10-minute break, and we'll spend a little bit of time going over the midterm and field any questions you have, and then we'll call it a day. Okay? So I'll see you guys at 11 o'clock.